Greetings, everybody. So today we are going to discuss about circulation, and here the topic which is important to be discussed. So this is a topic which we are going to have a one shot of. We're going to take the one shot of the topic which is circulation. Here we are going to discuss each and everything. So uh, first part, coming to what is circulation. Circulation basically it is the transport. It is a mechanism in which we transport oxygen to different cells of the body, nutrients to different cells of the body, as well as in return exchange the waste materials and metabolic waste from the different cells of the body. Now, transport occurring within simple living organisms such as amoeba can be, facilitated by simply diffusion. Likewise, when it comes to other organisms such as Hydra can also have transport through diffusion. But those organisms which are having a complex multicellular structure, they require a circulatory system through which they can distribute food, oxygen in different organs of the body and collect carbon dioxide and toxic materials which are produced due to the metabolic activity of the cell. So in this case, the system which is involved in this exchange of materials and transport of materials is known as transport system or the circulatory system. What are the general characteristics of a circulatory system? There are three major characteristics of circulatory system. Number one includes the medium in which the transport occurs. Now that medium is to be fluid. Uh, in humans, that fluid is in two forms. One is the blood, and second is the lymph. When it comes to blood, um, it's going to have a very big part we are going to discuss here. And when it comes to the second part, which is second characteristic of a circulatory system, it is the system, or in other words, it is the um, vessels in which the fluid is carried or the medium is present. Now, these vessels can be blood vessels as well as lymphatic vessels, which we are going to discuss. In humans, there are two types of systems: a circulatory system, lymphatic, and circulatory system. These are the two transport systems. When it comes to third part, it is a pump. Pump means an organ which is responsible for pumping the fluid throughout the body, and that organ is heart. Basically, it is a modified blood vessels. Why is that so? That is because heart um, during the embryonic development is nothing else but a blood vessel which is two or many blood vessels which are conjugated with each other in order to produce a complex, a very thick muscular blood vessel. So that is known as heart, and that is why we call it as a modified blood vessel. Now it is of two types: open type circulation, closed type circulation. Open type circulation was first seen in arthropods. Closed type circulation was first seen in annelida. Moreover, some basic characteristics in open type circulation. Open type circulation is the one in which there is no involvement of blood vessels, as well as there is no distinction between blood and the tissue fluid, interstitial fluid, or intercellular fluid. Likewise, closed type circulation is the one in which there are blood vessels involved, as well as there is a distinction between the blood as well as the interstitial fluid, tissue fluid, or intercellular fluid. And now some basic characteristics: blood is colorless due to the absence of hemoglobin in open type circulation. Now it's not necessary that every time blood will be colorless, because in hemocyanin there is also open type circulation, but the blood is blue because of um mollusca. Sorry, in mollusca the blood is blue because of presence of hemocyanin. Even though they are having open type circulation, this is because they are not having hemoglobin but are having hemocyanin, but still it is an open type circulation. Likewise, when it comes to other part, the blood flows chamber to chamber present in the body and supplies the food of the organs present in that chamber. Now, um, these the blood is fluid into the chambers of the body, and this chamber is known as hemocoel or cavity, which is known as hemocoel. And that blood which is flowing, actually it is not called blood because there is no distinction between blood and tissue fluid, so we call it as hemolymph. And that hemolymph either takes the color of the pigment which is present within the hemolymph, or most of the time it is colorless. The blood does not carry oxygen. There is no carrier of oxygen in it. And when it comes to number 4 point, the blood is called hemolymph. As previously told, the blood capillaries are absent, and blood cavity is called hemocoel. And this is all about. open type circulation. When it comes to closed type circulation, they have basic characteristics found in annelida, fishes, reptiles, mammals, and these are just opposite of open types. Such that here the blood is colorless, but in closed type circulation, blood is red due to the presence of hemoglobin, as well as the blood flows in blood capillaries in the entire body, particularly blood vessels, and blood supplies the food, oxygen to the parts of the body and collect toxic materials in return, and to take these to kidneys and lungs to remove for the removal of body, unnecessary stuff. Moreover, when it comes to the closed type circulatory system, it's also mentioned in the diagram. Here it is divided into two types: the single circuit circulation, which is found in fishes, and double circuit circulation, which is found in mammals, reptiles, and amphibians. Now, when it comes to single circuit circulation and single circuit circulation, now single circuit circulation is in such a manner that here it is a characteristic of animals having two chambers, two chambered heart present, which is basically consisted of auricle and ventricles. The blood is connected from different parts of the body and poured into the auricle through the sinus venosus, and that sinus venosus basically is a part um in which from where the blood is entered into the auricles, and after the contraction of auricles, by the way, this is the only heart which does not receive oxygenated blood. The single circuit heart does not receive oxygenated blood, such that the system can be explained in such a manner that, let's suppose this is sinus venosus, it is coming into the sinus venosus, which is coming into the two chambered heart. These are the two chambers. Let's suppose this is auricle, this is ventricle, this is sinus venosus, and here when blood comes into the auricle through the sinus venosus, that blood coming into the auricle now upon the contraction of auricle goes into the ventricles, and upon the contraction of ventricle is now taken outside into an artery which is known as conus arteriosus. A blood vessel known as conus arteriosus or bulbous arteriosus. Conus arteriosus, bulbous arteriosus. Some books also mention that there are 4 to 5 conus arteriosus or bulbous arteriosus. That's conus arteriosus further. um this is basically a proximal swollen part. Conus bulbous arteriosus, it is a proximal swollen part of ventral aorta, such that when this swollen part, after being passed into, after the blood being passed into the this swollen part, it directly goes inside the structure which is known as the ventral aorta. Ventral aorta. And from the ventral aorta, the blood leaves directly, and from the ventral aorta, the blood leads into the gills. Now, gills, respiratory organs. So, from the gills, the blood gets oxygenated, and once it is oxygenated, it is then now transferred into the dorsal aorta. Dorsal aorta is the first part which receives the oxygenated blood, and once the dorsal aorta receives it, the dorsal aorta now transfers it to different parts of the body by dividing into several arteries. We are not going to discuss that here. It divides into different parts of the body. Different parts of the body, it is divided into different parts of the body, dorsal aorta, and once it is transferred into different parts of the body, the blood again becomes deoxygenated and is then transferred through the different um, veins back into the sinus venosus, and from the sinus venosus it is then back transferred into the heart. So this is a single circuit circulation. And when it comes to the supply of heart muscle supply is similar, just the difference come when the blood is oxygenated from the dorsal aorta. Oxygenated blood from the dorsal aorta arches into a coronary artery, and from the coronary artery it is then transferred to the heart muscles. It is then transferred to the heart muscles through the coronary artery. If it is being transferred to the heart muscles, and once the heart muscles receive the blood becomes deoxygenated, it is left behind through the coronary veins from the heart muscles, and then transferred into the sinus venosus um, as the deoxygenated blood. So this is a basic concept of it. Let's study what it is about. The deoxygenated blood is taken to gills by means of conus arteriosus. Bulbous arteriosus generates the passage through the mouth, and exchange of gases takes place, and here contains deoxygenated blood without any um, hence termed as venous blood. So the heart basically contains the deoxygenated blood or the venous blood. Moreover, when it comes to double circuit circulation, um, it is a circulation in which either a three-chambered or four-chambered heart is involved. Such that if we are talking about a three-chambered heart, that three-chambered heart will be having the auricles, two of the auricles, two auricles would be present, left auricle, right auricle. I'm talking about the heart of amphibians. Amphibians are having three-chambered heart, so they have three-chambered heart, and here there is right auricle, left auricle, right, and left auricle, and there is a ventricle which is present downwards. Now here, the system is in such a manner that here the blood comes into the right auricle through an artery, uh, through a vein which is known as sinus venosus once again. This is not as simple as it seems. Here, first and foremost, there are two superior vena cava coming. I'm talking about the heart of amphibian once again. Two superior vena cava and one inferior vena cava. Two superior vena cava and one inferior vena cava uh, leads the deoxygenated blood into the sinus venosus. Superior vena cava, also known as precaval, and inferior vena cava postcaval, transfers the blood into the sinus venosus, and from the sinus venosus it is then pumped into the right auricle. Right, at the same moment, the oxygenated blood from the pulmonary vein is then transport from the lungs is then transported into the left auricle upon the contraction, upon the relaxation, sorry. And now, once one chamber is filled with oxygenated, other chamber is filled with deoxygenated blood, both now both chambers contract simultaneously, as a result of which the blood from the right auricle and left auricle is then transferred into the ventricle upon the contraction, as a result of which the oxygenated as well as the deoxygenated blood is um, mixed up. So as there is mixing or intermingling of the deoxygenated and oxygenated blood, it is known as an incomplete double circuit circulation. Incomplete double circuit circulation. And here this mixed blood, um, due to incomplete double circuit circulation, is kept within the auricle, and from the auricle there is arisal of an aorta which is known as truncus arteriosus or conus arteriosus, which further uh, branches into six arteries: two systemic arch, two carotid arch, and two pulmocutaneous arches. So these are the number of arches in which it is divided, and this is a basic formula. And here, incomplete double circuit circulation, it is so called because there is mixing of oxygenated and deoxygenated blood in the ventricle. So this is the reason as well. In the amphibians, it is known as incomplete double circuit circulation. As well as the you may see that the auricle and left auricle is separated from each other. These are separated from each other through an septum. This is a septum which is known as the interarterial septum. Apart from which, when it comes to the heart of reptiles are basically having the same mechanism. A difference only comes when their heart is partially partitioned, such that let me just try to explain. Here we are having the so here there is a structure such that here we are having the heart, and here there's right atrium, there is a left atrium, and the ventricle is also you know a single, but that ventricle is partially partitioned, such that it is partially partitioned through some sort of membrane, and here there is a gap inside it, so it is partially partitioned, and that region in which the blood from the right atrium comes inside this region in which the deoxygenated blood is arriving in the ventricle is known as cavum pulmonale, and from the cavum pulmonale the blood is then transferred into the pulmonary artery into the lungs. Likewise, left atrium carries the deoxygenated blood into a partially partitioned chamber, which is known as cavum arteriosum. And from the cavum arteriosum, the blood is transferred into the conus arteriosus, or a truncus arteriosus. And truncus arteriosus basically transfers the blood into two systemic arches, right and left systemic arches. So this is how the blood is transferred. One exceptional case, that crocodile it belongs to the class reptilia but has four-chambered heart. And one more thing, that here this partially chambered heart, um, partially chambered heart is uh, leading to that sort of region, which is known as cavum pulmonale and cavum arteriosum respectively. And rest, there are also superior vena cava, inferior vena cava, which are same. And instead of formation of six arches, there is only formation of two arches, systemic left and right systemic arch from the conus arteriosum or truncus arteriosum. And here the mechanism is almost similar. Now coming to the next point, that when it comes to complete, it is also an incomplete double circuit circulation. Moreover, when it comes to complete double circuit circulation, it occurs in humans, which we are going to study in detail. Here there is right atrium, right ventricle, left atrium, left ventricle. They have interarterial as well as interventricular septum. This is interarterial septum, this is interventricular septum, such that there is right atrium, there is left atrium, left atrium, there is right ventricle, there's left ventricle, right. So this is how the structure is formed. Now the atria are completely separated from the ventricles through a groove or depression, which is known as atrioventricular groove. That atrioventricular groove, basically it is made up of it is made up of some fibers, as well as some ring-like structures, four ring-like structures, which are known as annulus fibrosus. These four ring-like structures are known as annulus fibrosus. Each ring is known as annulus fibrosus, which connected with the cardiac skeleton or the myocardium. Four rings, annulus fibrosus, and each annulus fibrosus is connected to each of the valve of heart. One more thing I forget to mention is that in the single circuit circulation of fishes, there are also valves present throughout the vessels, such that these valves prevent the backward flow of blood. Likewise, in amphibians and reptilia, there is same case. So here, coming to this point, here we are having annulus fibrosus, which is each connected with each of the valve, and therefore valves and some other valves are present as well. Moreover, when it comes to the blood supply, we are going to discuss it here. Here this is on complete double circuit circulation. Heart of man, first and foremost, it is a pumping organ. It is the most powerful or dynamic organ within the body. It helps to circulate the blood throughout the blood. The blood is body sorry the blood is The blood is red in color due to the presence of hemoglobin. The heart is a muscular, hollow, conical shaped organ but present in the cavity of thorax. Hence, it is known as pericardial cavity. It is covered by a prominent covering, which is known as pericardium, between the lungs, with one third of it situated at the right and towards the left, what's on the left of the media sternum. Medium breeze being behind sternum. This is known as media sternum position. So that it is behind the sternum towards the right side, such towards the left side. One third of it is towards the right side, and two-third on the left side, as well as it is between the ribs, and between which rib, between fifth and sixth rib, as well as it is mesodermal in origin, apart from which it is about the size of one's fist, and it is dimensions it is around 12 centimeter long, 9 centimeter wide, and 6 centimeter in thickness. This is moreover, it has conical shape, and apart from which we are having some basic details, such as how the layers of heart are arranged. The heart walls consist of three layers enclosed in pericardium. It is thick layer which covers the heart and roots major vessels, which consists of an outer fibrous layer, which is made up of fibrous pericardium, and inner double serous membrane layer called serous pericardium, right? As well as this layer is in such a manner that here the fibrous pericardium consists of thick fibrous connective tissues and forms the borders of the middle media sternum. on the borders of the middle mediastinum. Serous pericardium, it is very close to the heart and its two layers. A small amount of serous pericardial fluid is present. It lubricates the layers and prevents friction during the heart contractions. It also provides mechanical support as a basic function of the pericardium. Moreover, uh, let me just try to explain it over here. As you can see, the heart layer is in such a manner. This is heart, and the outermost layer is on a fibrous pericardium, which is in contact with the thoracic cavity, the mediastinum region, mediastinal lining, which is known as fibrous pericardium. And after that, we are having the serous pericardium. It is a double layer. It is the fibrous pericardium made up of thick fibrous connective tissues, and serous pericardium consists of serous layer, membranous layer, and it is of two layers. First, outer is the parietal layer, which is in contact with the fibrous pericardium, and the inner one is a visceral layer which is in contact with the epicardium of the heart, and that is basically uh, you know composed of serous layers. So it is generally made up of mesothelium and some connective tissues, and space between both of these is known as the pericardial cavity. Space between both of these is known as pericardial cavity, and pericardial cavity uh, is filled with a fluid which is known as pericardial fluid, which has a number of functions, such as it acts as a lubricant to prevent friction, as well as it causes to act as it causes to act as a lubricant first and foremost. Secondly, it prevents overextension of, or due to, because if overextension occurs, the heart can be overfilled with blood, as well as it prevents shocks and mechanical injuries from the heart. So this is a function. After that, after the pericardium, we are after the visceral, inner visceral pericardium, we are having another layer present, which is known as the epicardium. That epicardium basically, it is the outermost layer, and it is made up of mesothelium as well as connective tissues. Sometimes it is also said that uh, inner serous pericardium, um, the inner visceral serous pericardium forms the epicardium. Well, it can be said true to some extent. And apart from that, epicardium, it is the outer layer of heart and is formed by the visceral layer of the serous pericardium, and it is basically in contact with the serous inner visceral serous pericardium. Some books also mention that the inner visceral serous pericardium is itself epicardium, but according to our book, it's not the case. And here, when it comes to myocardium, myocardium is the second layer, which is myocardium is the second layer, which is basically the thickest layer, and that is also known as a cardiac skeleton. It is also known as a cardiac skeleton of heart. It is composed of cardiac muscle fibers, as well as elastic tissues and conducting system, such that it is a conducting system of cardiac musculature. Over that cardiac musculature is basically known as nodal tissue. These nodal tissue are SA node, sinoatrial wall, sinoatrial node, and atrioventricular node. These are the two nodes present. We are going to discuss what are these as well in the conducting system. And here, these are basically, myocardium is made up of thick muscular fibers, and these muscular fibers have several extensions as well, due to which they form different structures such as papillary muscles or musculi papillaris, as well as it causes to form another structure within the myocardium, due to its extension, which is known as uh, the different muscles are there. So we are going to discuss in it later. And here, one more thing, that myocardium, it is the thickest layer. It is known as cardiac skeleton, as well as its composition basically, it is made up of troponin, myosin, tropomyosin, as well as actin, as well as dystrophin. These are the muscle fiber cells which are present here. And apart from that, um these muscle fibers compose line, and here the T system. We are going to discuss that as well. That the T system is present on the A1 on the Z line instead of A1 line, as it is in the case of skeletal muscles. Apart from that, when it comes to endocardium, it is endocardium, it is the in middle concentric layer, such that it is a subendocardial layer, and the rest of the heart is composed mainly of subepicardial and endocardial layers, right? So it is a concentric layer, subendocardial layer. The rest of the heart is composed mainly of subepicardial, subendocardial layer. One more thing, that here the endocardium has several extensions. It has several extensions, which are known as the walls. The extensions of endocardium basically forms the bicuspid and tricuspid walls, as a result of which we are able to see all of these bicuspid and tricuspid walls, which are in actual the extensions of the endocardium. Endocardium itself is made up of uh, endothelium cells, of endothelial cells, as well as a thin basement membrane, which is made up of loose connective tissues. One more thing, that epicardium is connected with the myocardium with the help of connective tissues, and myocardium also has some sort of connective tissues which help it in forming its cardiac muscular structure. And apart from that, when it comes to other part, let us discuss about the structure of heart. Structure of heart is in such a manner that here the heart of man consists of four chambers: right atrium, right auricle, left atrium, left auricle, right ventricle, left ventricle, such that blood both are separated by intra-arterial septum. There are two openings, one pre-cable and other post-cable, which open separately and brings deoxygenated blood from all parts of the body. The left atrium, two pulmonary veins opened by the separate apertures through which bring oxygenated blood from the lungs. In this way, in the right atrium, deoxygenated blood, and in the left atrium, oxygenated blood is collected, such that when oxygenated blood is collected in the left atrium, it is known as, the blood is being transferred from pulmonary circuit, so it is known as pulmonary circuit. And when it is being transferred into the body, so it is known as systemic circuit, we are going to discuss. And here, right atrium is deoxygenated, left atrium oxygenated. Several walls are present over here as well, such as uh, Haversian wall, Thebesian wall, and falciform fold. These are walls present. Haversian wall is present on the inferior vena cava, Thebesian wall present on the coronary sinus, and Eustachian wall present on the Eustachian wall, which is present on the superior vena cava. Moreover, the moreover, when it comes to the other structure I would like to highlight over here is that uh, the coronary sinus, it is basically um, a sinus. It is a blood vessel which directly supplies the deoxygenated blood from the heart muscles directly to the heart's right atrium. And this is how actually the supply occurs. And when it comes to right and left ventricles, they occur in such a manner that two ventricles from the lower part of the heart are separated from each other by interventricular septum. The right ventricle opens into an aperture into the right ventricle through right atrium, opens into right ventricle through right auriculoventricular aperture. This aperture is guided by an atrioventricular wall, which is known as tricuspid wall, which is having three cusps or flaps, and these flaps basically are membranous extensions of endocardium, which allow the blood to flow from the right atrium into the right ventricle, but not in the backward direction. The left atrium opens into the left ventricle by an aperture, which is known as left auriculoventricular aperture, which is guided by a bicuspid valve or mitral valve, which allows the flow of blood from the left atrium into the left ventricle, but not in the backward direction. Internally, the walls of ventricles are raised into conical processes called papillary muscles. Now, these papillary muscles or musculi papillaris are basically those muscles which are typically, you know, these are also extensions of myocardium, and these are connected to the flaps of the tricuspid and bicuspid wall through inelastic thick tendons, which are known as chordae tendini. And these chordae tendini basically lead musculi papillaris to control the closing and opening of valves the during the cardiac cycle, as well as when it comes to as well as three valves are present on the three papillary muscles are attached to tricuspid wall. Two papillary muscles are present in the attached to the bicuspid wall. Moreover, these are attached to the papillary muscles by threads known as chordae tendineae. These threads control the movement of walls from the right ventricle. Pulmonary aorta arises. Its opening has three walls called semilinear walls. Now here the pulmonary aorta, when the blood moves from the right ventricle into the lungs through the pulmonary aorta. That pulmonary aorta is having a wall present, which is known as particle. Pulmonary aorta. Pulmonary aorta, which is having a wall present known as semilunar walls, which looks like half moon, due to which we call it as a semilunar wall. And it is membranous again. It is precus, but as it looks like half moon, we call it as a semilunar wall. When it is due to presence on pulmonary aorta, we now also call it as pulmonary wall. One more thing, these walls permit the flow of blood from the ventricles into the pulmonary aorta, but not in the backward direction. From the left ventricle, an arch called aortic arch and systemic arch arises at its opening as semilunar wall, which allow the outflow of blood from the ventricles into the aortic arch, but not in the backward direction. Here, there is also present a wall known as aortic wall, uh, which is basically a semilunar wall. After that, phases of heartbeat is in such a manner from all parts of the body. The deoxygenated blood is collected by precaval and postcaval, which opens into the right atrium. In the left atrium, oxygenated blood comes from the lungs through pulmonary veins, which when the atria are full of blood, when the atria are full of blood, they contract at the same time, and the blood comes into the ventricles. And at that moment, this is known as the atrial systole. Systole, the atrial systole, the right, which is lasts for around 0.3 to 0.4 seconds, which lasts for around 0.1 second. Sorry, 0.1 second. We're going to calculate it as well. The cardiac cycle, which basically lasts for 0.8 seconds. uh, Cardiac cycle means one complete flow, contraction and relaxation of one complete cycle of heart. And here, when the atria are full of blood, they contract at the same time, and the blood comes into the ventricles. The right ventricle receives the deoxygenated blood, and the left ventricle receives the oxygenated blood. When the ventricles are filled with blood, they also contract at the same time. The bicuspid and tricuspid valve prevent the backward flow of blood into the atria auricles. When the ventricles contract the chordae tendineae, it do not allow the valves to push back into the atria, right? So the bicuspid and tricuspid valve prevent the backward flow, and the chordae tendineae does not allow valves to be pushed back into the atria. And when ventricles contract, the semilunar valves allows the blood to flow into the pulmonary aortic arch and pulmonary artery. From the right ventricle, the blood comes into the pulmonary artery, which carries this deoxygenated blood to the lungs, where oxygenation takes place. From the left ventricle, the blood flows into the aortic arch, systemic arch, which supplies the oxygenated blood to different parts of the body. In this way, the two systems of circulation work in the body of man. One is pulmonary circulation, pulmonary circuit, which runs from blood transferring towards the lungs and being transferred back away from the lungs, and systemic circulation, in which blood is being taken from the different parts of the body and then is being transferred to different parts of the body. A common MCQ is here, which is the largest vein. Largest vein is inferior vena cava, and largest artery is systemic aorta. The second circulation is systemic circulation, which takes place between the heart and other parts of the body through the systemic or aortic arch. This is known as complete double circuit circulation, right? And this is how we basically uh, write it. But when it comes to several phases, the phase the in which the blood present in the right atrium and left atrium basically releases from the atria to the ventricles. Due to the contraction, it causes to have a time period of 0.1 second in which the blood is then transferred. Now, it's not the exact time. Of course, there can be some variations. 0.1 second, as a result of which this condition in which the blood is contracted when the atria are contracted. This causes as the atria contracted within this period, atrial systole. Systole basically refers to contraction, and diastole to relaxation. Systole basically, as contraction occurs in the atria, it causes the to first and foremost, there's already a lot of pressure when the blood is filled within the atria. And on the other hand side, if we are applying more pressure through contraction on the atria, this will cause the valves which are present, uh, tricuspid and bicuspid valve, to resist, to uh, endure the pressure, and there will be a time when they will burst open. As a result of which, when pressure is ultimately increased, the uh, valves will ultimately open up and thus release the blood into the ventricles. This period is known as atrial systole, which lasts for around 0.1 second. After that, this releases the blood into the ventricles, into the ventricles, as a result of which previously the pressure of ventricles was less, but as the blood was transferred into the ventricles, it caused the ventricles to have their pressure over the uh, valves, which are known as the semilinear valves, increased, as a result of which semilinear valves close up. Moreover, as this process occurs, uh, as the blood is completely filled, as the ventricles are completely filled with the blood, the second phase occurs. In the second phase, we are having the contraction of ventricles, and that is known as ventricular systole. Such that in ventricular systole, when atrial systole is finished, the tricuspid, bicuspid valves are closed. It causes it causes a condition which is known as the ventricular systole, contraction of ventricles, as a result of which the blood is then transferred from the ventricles into the aorta, pulmonary and systemic aorta, by pushing or forcing the walls, semilinear walls, to open up. And in that case, first and foremost, there are three conditions causing it. First condition is the blood present within the ventricles, which causes the extension of ventricles, distension of, sorry, ventricles, as well as uh, there is the closed wall. As the walls are closed, it means that blood will cause to flow back, and this blood will which will be causing to flow back, that will also exert a pressure on the walls. And third condition is the contraction itself, as there will be contraction of ventricles, and this contraction of ventricles will lead the deoxygenated blood to move from the ventricles into the aorta by pushing open the semilinear wall through a period which is known as ventricular systole. And that ventricular systole lasts for around zero point 0.3 seconds. 0.3 seconds. During the period of ventricular systole, the atrial systole, during the period of ventricular systole, the atria remains relaxed. So the during the period of ventricular systole, there is atrial diastole, which means atria remains relaxed, and at that moment, the atrial systole lies for around 0.3 seconds, because 0.3 second is the ventricular systole, and here 0.3 second is the atrial diastole. Now we are going to add another quantity, because of course it's not the when ventricular diastole will be relaxing, there will also be relaxation of atria. So at that moment, that period will last for around 0.4 second. And when we are supposed to add both of these values, we'll get the total value of atrial diastole alone as 0.7 seconds, which means the atrial systole only occurs for 0.1 second, right? And after that, when the blood is transferred, basically when the blood is transferred in such a manner that here, as you can see, let me just try to draw it here. When the blood will be transferred, let's suppose these valves are closed. These valves are closed. When the blood will be transferred into the ventricles and the valves will ultimately close, this will cause the blood which is coming from this side. Let's suppose blood has come, and now it causes the the walls to close up. As the walls close, the blood causes to perform a thrust. A thrust occurs of blood, due to which the blood moves in the backward direction towards the closed valves. As it moves in the backward direction towards the closed valve, this closing of valves, due to which the blood is thirsted over the tricuspid and bicuspid valve, produces a sound which is known as first heard sound, which is known as lub, which is due to the ventricular, which is due to the ventricular contraction. Ventricular contraction, as contraction occurs, some of the blood is thirsted back, due to which it will cause the blood to have producing a sound which is known as lub dub, right? So lub, basically it is having high amplitude, and dub is a sound. Lub is a sound having high amplitude, and dub is a sound which is having high pitch or high frequency. So these are the things. And a dub is a sound which is created when the blood here, when blood is released through the ventricles. When blood is released through the ventricles, it will cause the as the blood is released from the ventricles and the semilunar walls are closed. After the ventricular systole, the semilunar walls are closed. It will cause some of the blood, some of the blood present within the pulmonary and systemic aorta to move back into the ventricles, due to which uh, even though due to which the blood will tend to move back, but the semilunar walls since are closed, it will cause the semilunar walls, since the semilunar walls are closed, it will cause to again collide the blood with the semilunar walls, as a result of which it will cause to produce a second heart sound, which is known as dub, lub dub. There are two other sounds as well. Two other heart sounds are there, known as S2 and these are S1, S2, and S3 and S4 are there. S1, S2 basically it is due to uh, we are having S3. S3 basically it is caused due to the ventricular contraction, and S4 is caused due to the arterial filling. But these are two small sounds, so we usually don't measure it. We can't hear them, usually in a heartbeat. And after that, when the after the ventricular systole, when the semilunar walls are closed, the process which occurs now is when is diastole. Diastole here occurs of both atria as well as ventricle, such that the heart uh, muscles of both atria and ventricle remain relaxed, and this period is known as diastole, in which there is filling of blood. Filling of blood occurs. Filling of blood in the atria and atria occur, and that filling of blood in the atria produces a sound which is known as S3 sound, which is quite short sound, right? And it lasts for around 0.4 seconds. And apart from that, when it comes to the cardiac cycle, it is also similar. The heart muscles are auto-excitable. They are myogenic, such that they are on, they generate their response automatically without any need of any response, and uh, uh, any need of and stimuli from the nervous system. So they are myogenic. They generate their own cardiac contraction relaxation. Their rhythmical contraction starts from within the muscle without any external stimuli. Between the two contractions, there is a short pause. The sequence of events that takes place during the completion of heartbeat is called cardiac cycle. So this is the cardiac cycle in which atrial systole, ventricular systole, and atrial diastole occurs. And here this process in which first was the atrial systole, which lasted for 0.1 second. Then there was ventricular systole, which lasted for 0.3 second. After which there was what do we call it? The diastole. When you're going to add these values, we'll get our answer 0 0.8, which is the total time period for a single cardiac cycle. And the resting period of heart chambers is diastole, working as systole. And here the right ventricle and ventricle is termed as atrial systole and ventricular systole. The amount of blood pumped by the heart by the pumped per minute by the left ventricle is called cardiac output. And apart from that, when it comes to heartbeat, the rhythmic contraction of heart chambers is known as heartbeat. Heart contracts rhythmically, and which is 72 times per minute in a healthy person, and below 60 times it is known as bradycardia, and above 60 times, above 100 times it is known as tachycardia. And the ventricular valve is followed by ventricular diastole, forcing the aortic valve to close it. It produces a sound which is known as dub. One complete heartbeat consists of one systole and one diastole. It lasts for about 0.8 seconds. Any defect in heart is known as murmur, which is detectable as a hissing sound. It is murmur which is detectable as a hissing sound. Now, the diagram is also mentioned how does the mechanism actually works. And here, as you can see now, we are moving forward. SA node. SA node, the contraction of the heart is due to the stimulus which originates at SA node, which is situated near the opening of precabal in the right atrium. It is the remains of sinus venosus having the nerve ending in it. So it is basically the remain of sinus venosus having the nerve ending in it. By the way, important point that here systemic arch. There are two systemic arches, left and right, in reptiles, but in birds during embryonic development, the left systemic arch disappears and right systemic arch remains, due to which they have a right systemic arch throughout their life. Mammals have opposite, that they have right systemic arch disappeared and left systemic arch appeared during throughout their lifetime, during the embryonic development, due to which it causes to transfer blood through the left systemic arch. This is the difference between the bird and mammal uh, systemic circulation. And apart from that, since AV SA node initiates the heartbeat, it is known as pacemaker. AV node, it is situated below the SA node in the right atrium. It is excited by the above by the wave produced by the SA node. Here, the impulses are delayed for about 0.1 second, which ensures that atria will contract first and empty completely before the ventricles are contracted, which reaches here where the muscles known as Purkinje fibers, and that these Purkinje fibers lead uh, first and foremost, the bundle of his arises in such a manner that, let me just see if there's a diagram available over here. Uh, there are several fibers here. Let me just try to draw the structure. Here, the heart walls are separated from each other. By the way, left ventricle is the thickest chamber, and atria are thicker than ventricles are thicker than atria. Moreover, the left ventricle is three times thicker than right ventricle. Moreover, the right atrium and left atrium basically are arranged in such a manner that right atrium is a bit bigger than the right left atrium. Reason behind it is that it receives the supply of the entire body, so it needs to be bigger. Apart from which, when it comes to the left ventricle is the largest, thickest chamber, but it is smaller. It is having a smaller cavity as compared to the right ventricle. Reason behind it is that the interventricular septum here present is, it's not a straight septum. It is a bit obliquely transversed towards the right side. It is obliquely transversed towards the right side, due to which the right ventricle is smaller, left ventricle is bigger, but the cavity of right ventricle is bigger and left ventricle is smaller. Now, coming to the point, here we are having the SA node. SA node is present towards the right side, towards the left side of the right atrium. Towards the left side of the right atrium, we are having a nodal structure made up of nodal tissues known as the SA node, and that SA node basically uh, carries some signals through bundle towards through several internodal tissues. There are three internodal tissues of Bankman, international internodal tissues of Wank, and internodal tissues of Thuritel. These are three internodal tissues which are transferred, from which uh, these internodal tissues transfer the impulses. Once the impulses are transferred uh, to contract the heart, these impulses are now transferred through the internodal tissues towards the AV node, and AV node, atrioventricular node, is from where it is transferred. And still there are some doubts going on that whether internodal tissues are truly capable of doing that, because the internodal tissues are separated from the ventricular muscles through the atrioventricular groove, as a result of which both muscles being separated has some doubt and confusion going on. Apart from that, there is a projection. There is the only system of muscular projections which connects the atrial muscles with ventricular muscles. Although both of these muscles are completely partitioned through atrioventricular groove, there is only one muscular projection which connects atrial and ventricular muscles, which is known as bundle of his. Bundle of his rises from here, from the SA node to the AV node, and from the AV node it arises, and it arises in the form of, first and foremost, AV node has several bundle of his arising from it, which generates the impulse such that the bundle of his goes straight, and the left ventricle, you know right atrium being near to the heart, the sorry right ventricle being near to the bundle of his, first divides the bundle of his into the left ventricle, right? After that, when it comes to the come to rest of the bundle of his, rest of the bundle of his goes towards the right atrium. So this is how it actually occurs. um, Let me just try to draw it again. It's a bit messy. Right. So here, as you can see, the structure will be arranged in such a manner. There are several a bundle of his arises, such that this is a bundle of his arising. First and foremost, it goes straight and then branches into the left ventricle first, because left ventricle is nearer to the bundle of his, and then it branches to the right atrium and continues towards the right atrium. From the bundle of his, there is arisal of several fibers which are known as Purkinje fibers. Purkinje fibers. These Purkinje fibers basically transmit the impulse towards uh, these Purkinje fibers are able to transfer the impulse towards the ventricles. And here, an interesting fact is that the Purkinje fibers can, they have a capability that they can generate myogenic stimulation, which means they can generate myogenic impulse, as well as they can generate neurogenic impulse, which means they can also generate neurogenic responses. And apart from that, when it comes to the his bundle of his, it has also its own heartbeat. Basically, SA node as the pacemaker of heart has its heart maintains a major heartbeat of heart, which is 0.8 second. But when it comes to AV node can also generate its own heartbeat, but that would be considered as an irregular heartbeat in case if SA node is not working. Then at that point, this irregular heartbeat would be considered as cardiac arrhythmia. Cardiac arrhythmia. And if the heart is located a bit on the right side, it is known as cardiac dexter. This is a condition. Moreover, uh, apart from that, when it comes to the system, here the the conducting system transfers the impulses from SN node to AV node during the cardiac cycle, and from the AV node to bundle of his through the Purkinje fibers, and bundle of his as well as the Purkinje fibers as well as the AV node as well as the SN node. All of these are having their own impulses, such that when it comes to SN node, it is having its own impulse 0.8 sec. It causes and generates an impulse of 72 times per minute, and AV node has an impulse of around 30 to 40 times per minute. And when it comes to the bundle of his around 25 to 30 times per minute, and when it comes to Purkinje fibers, around 20 to 25 times per minute. Right. After that, artificial pacemaker, it is a device which supplies electrical impulses to the heart to maintain the heartbeat at regular rate. It is used when SN node is not functioning properly. And electrocardiogram, the electrical impulses that pass through the cardiac muscles during the cardiac cycle produces electrical current that are conducted through body fluids through the body surface, where the currents can be detected by electrode potentials by electrodes placed on the skin and recorded as electrocardiogram. So the electrocardiogram ECG machine. And here you can see there are several phases of electrocardiogram, such that we are having a mechanism of it is in such a manner that here, when it comes to electrocardiogram, it helps to check electrical changes in the form of depolarization and repolarization. When there's accumulation of sodium ions, this is known as depolarization in the heart. Sodium ions cause depolarization, and potassium ions cause repolarization. And here, what actually occurs in depolarization is a depolarize means charge is being finished, which means impulses are being finished. When it comes to repolarization, which means again the impulses will be generated in order to form in order to generate the systole or diastolic response. Now, coming to the point, how does it actually occur? First and foremost, there's showcase of depolarization of the RTL fibers of the SA node. First and foremost, the depolarization of the ATL fibers of the SA node produces the P waves. Now, P wave basically it is the wave which is generated as a which is generated before the ATL systole in order to contact the systole, and that basically is due to the sodium ions depolarization. The ventricles of the heart are in diastole during the expression of this wave on the ECG recording. PR interval, it is the period from the start of the P wave to the beginning of the QRS complex. This interval indicates the amount of time required for the SA node depolarization to reach the ventricles. And this interval indicates this interval. Now, this is known as P waves, and PR interval. It is the interval in which the atrial systole occurs, and it is the period from the start of the P wave to the beginning of the QRS complex. It indicates the amount of time required for the SNO depolarization to reach the ventricles, and once it actually occurs, it causes throughout the period, the impulses are being generated from SNO to the ventricles. This period will lead to a condition which is known as atrial systole until the time when the impulses are left behind. When the impulses are reached over the ventricles, the atrial systole finishes, atrial diastole starts, and ventricular systole starts ultimately. And this period rise for around, let me just write it. Here we are having this period P wave, and P wave, this complete period lasts for around zero point 0.1 second, zero point 0.1 to 0.15 second. After that, this is the ventricular the period which is before the ventricular diastole, period which is before the ventricular systole, sorry, and it begins as a short downward deflection. Q continues as a sharp upward spike and ends as a downward deflection. The QRS complex basically indicates the depolarization of the ventricles during this interval, such that when the ventricles receive that impulse, it causes depolarization to, in a way that it generates electric response in the form of a depolarization towards the ventricles through the bundle of his and Purkinje fibers, and the ventricles are in systole upon accepting that uh, electrical impulse, and blood is being ejected from the heart once ventricular systole occurs. And in this case, this is at a very high peak, high uh, frequency. And here ST segment, it is a segment in which the actual ventricular systole is occurring. It is a time duration which represents the period between the complete of the ventricular depolarization and initiation of the repolarization, in which when the depolarization occurs, it leads to repolarization. And throughout till the time repolarization is occurring, the the period which is occurring here would be known as ventricular systole. After the T wave, it is produced by ventricular repolarization, and in the final phase of it, the normal ECG shows that heart is functioning normally. The second wave of the QRS complex occurs just before ventricular contraction, which is known as a repolarization, which indicates that after getting the repolarization signals of the potassium ions through the help of potassium ions, it causes to through the help of potassium ions, it basically causes to it basically causes to give us it causes to it, cau- it causes to give us the repolarization, such that here this repolarization leads to a condition repolarization repolarization basically leads to a condition in which it is the electrical impulse generated before uh, the diastole of both atria and ventricle. So this is a basic concept about ECG. And then when it comes to blood vessels are from where when the blood is released from the heart, its generally journey follows a route starting from aorta to arteries to arterioles and to capillaries, and then to venules to vein, and then vena cava, and finally back to the heart. This is a basic concept we have studied in junior classes. And here, when it comes to their layer, these are the layers of heart which we are going to discuss over here as well. And apart from that, let me just quickly open up all the files. Blood vessels, where we are going to discuss here about the blood vessels, such that here we're having three major types of blood vessels: arteries, veins, and capillaries. When it comes to their basic structure, uh, arteries and veins are having three major layers present within them, which is known as um, three major layers present over them, which is known as tunica intima, tunica media, and tunica externa. Such that when it comes to first and foremost arteries are the blood vessels which take blood away from the heart, and these parts of the present parts of the body, uh, these are present in different parts of the body. They divide into small branches called arteries, which are connected to the capillaries. The arteries control the flow of blood in the capillaries. Moreover, all the arteries carry oxygenated blood except pulmonary artery as well as the umbilical artery, but that is present in fetal stage. And the walls of arteries are composed of three layers. Outer layer, which is tunica externa, it is composed of connective tissues. And what type of connective tissues? These are made up of collagen fibers, as well as they resist the internal blood pressure. And tunica media, middle layer, it is composed of smooth elastic fibers or muscles. They are elastic, and by the contraction and relaxation, the cavity of arteries can be increased or decreased, which is known as vasodilation or vasoconstriction. The arteries are smaller cavities, have smaller cavities than veins, as well as they have larger blood pressure than veins. Um, Internal layer, tunica interna, it consists of endothelial cells. Um, Endothelium, simple endothelium with a basement membrane in case, and the anterior part and slightly thick walled balloon. The arteries are, sorry, expand slightly like thick walled balloon, which is on a vasodilation between heartbeats. They call recoil, helping pump the blood and maintain a steady flow through the smaller vessels. Smaller arteries enter into small vessels called arterioles, which play a major role in determination of the blood and determining flow of blood, which is distributed in within the different parts of the body. And here, when it comes to capillaries, capillaries are having a simple structure such that uh, these are the finest blood vessels of arteries. These are around 0.1 um, they are having just 7 to 9 micrometer in diameter, and around, these are microscopic blood vessels are the finest blood vessels of arteries and veins. These are thin walled and narrower, or are the tiniest than the arteries and veins only. These are 7 to 9 micrometer in diameter. The pressure within the capillary causes a continuous leakage or fluid from the blood plasma in the surrounding space, which is known as the interstitial space in which the interstitial fluid or intercellular fluid is formed. Moreover, they they make a close contact between the circulation and the tissue, so the supply of important material takes place easily by blood circulation. The capillaries are in contact within the cells and tissues, so the exchange of important materials between the tissue fluid and blood of capillaries takes place by diffusion. The walls of capillaries consist of simple layer, which is known as endothelium layer, endothelial cells through which the diffusion of the substances takes place, as well as they have a basement membrane as well, which is of loose connective tissues. The amount and the flow of blood in the capillaries are controlled by arteries. One more common thing, that the flow of the blood pressure within the capillaries is less than the blood pressure within the aorta. Reason behind it is that the aorta is having its total cross-sectional area is 25 centimeters square, and when it comes to capillaries, it is much smaller, because even though they are having the smallest smallest, what do we call it, a cavity, but since they are having more of the total cross-sectional area, such that if we are supposed to talk about a single aorta, let's suppose this is aorta, this is the diameter, right? And when it comes to capillaries, capillaries are arranged in such a manner that here these are tiny capillaries. This is its total cross-sectional area, which is around 25 centimeter square, sorry. And when it comes to capillaries basically, even though having less space in the cavity, but these capillaries are dispersed. Let's suppose the blood supply in this was 180 milliliter, right? On the other hand side, and blood is being transferred into the capillaries, the capillaries are further divided into smaller, you know branches, due to which its total cross sectional area of this entire network will be greater than the total cross sectional area of the single aorta, due to which we can say the surface area of the capillaries will be more than the surface area of the aorta, due to which the blood pressure within the capillaries would be greater. Blood pressure within the capillaries would be slower as compared to aorta. So blood pressure is higher in aorta, then it decreases aorta, then it decreases in arteries, then it decreases further in arterioles, where they further decrease into the capillaries, where there is a lowest blood pressure in our capillaries. After that, from the capillaries, the blood pressure starts to increase in the venules, then it starts to increase in the vena cava, veins first, and then it again increases. It increases the most in vena cava. So this is how the mechanism occurs. From the capillaries, almost all material can pass except the large plasma proteins, red blood cells, and platelets are unable to pass due to the large size. Rest all of the material is passed. Now, veins are the blood vessels that carry the blood, towards the heart, and they are thin-walled vessels present at the tissue region, joined to form the veins. And the veins have thin walls. It consists of three layers, and due to larger lumen, they have less blood pressure. The veins are provided with assembly in the wall to prevent the backward flow of blood, and these are non-pulsatile, such that we cannot hear the pulse because they do not have the capability to, they're having thin elastic fibers, or we can say they have thin tunica media, due to which, uh, uh, you know, they are unable to perform vasodilation and vasoconstriction. As a result of which, we cannot hear the pulsatility or the hearing of pulse in veins, but we can hear that in arteries. And when it comes to its outer layer, it consists of hard and fibrous connective tissues, collagen fibers. Again, it is similar, but difference come in middle layer, such that middle layer, which is the tunica media, is thinner as compared to arteries are having thicker tunica media, and veins are having thinner tunica media. Likewise, inner layer of it is made up of endothelial layer, and outer layer basically it is a bit of thicker, uh, and vasoconstriction, vasodilation in veins. Now, role of arterioles in vasodilation, vasoconstriction, such that the amount of blood flowing through a blood vessel can be regulated by contraction or relaxation of smooth muscles in the tunica media, and it occurs under the influence of nerves, hormones, and chemicals produced by nearby tissue. An important point: parasympathetic nervous system causes to decrease. Parasympathetic nervous system causes to decrease the heartbeat, and causes the mechanism of parasympathetic nervous system causes to decrease the heartbeat, as well as it causes a mechanism of vasoconstriction, while its empathetic nervous system causes you know increase in heartbeat, and it causes a mechanism of vasodilation. Apart from that, uh, an important hormone in the sympathetic nervous system is the sympathin hormone, which causes the release of norepinephrine and epinephrine, or noradrenaline or adrenaline, both collectively are known as sympathin, which causes to increase the heartbeat as well as cause vasodilation. Well, like when it comes to parasympathetic nervous system, it includes example of vagus nerve. Vagus nerve causes to release acetylcholine, as a result of which it decreases the heartbeat and cause vasodilation, constriction as well. Arterioles and contractile according to the requirement of the supply of tissues and organs, they are divided into vasodilation, vasoconstriction. In this type, the arteries in the skin expand and bring more blood to the skin capillaries to disperse excess heat to the outside and to maintain a proper internal temperature. It occurs in hot summer days when we are having high temperature, and this leads to a condition in which the heat from the blood vessels is then absorbed by the sweat glands, as a result of which the sweat is formed within the sweat glands, and thus it leads to release of you know uh, sweat from the skin. After the vasoconstriction, in this condition, arteries in the skin become constricted or narrow in response to cold. So the decrease of blood flow to the skin helps to minimize heat loss, therefore preserve a normal internal temperature. In extremely cold conditions, fingers and toes can become bitten, because the arteries supplying the extremities are constricted, as a result of which usually in cold conditions we are having the, what do we call it, fingers and toes frostbitten. Apart from which, when it comes to blood supply, role of precapillary sphincter. So in case of any sort of emergency, such that if there is, let's suppose if we talk about the arterial, when the blood enters from arterial into the capillaries, that's region in which the blood enters into the capillary is known as arteriolar capillary, and the region from where the blood goes outside is known as venular capillary. And when the blood enters from the arterioles into the arteriolar capillary, it basically causes to have a sphincter which is known as precapillary sphincter. The flow of blood in capillaries is regulated by tiny rings of smooth muscles known as precapillary sphincter, which surrounds the junction between the arterioles and the capillaries. These sphincters open and close in response to local changes that signal the need of nearby tissues. For example, accumulation of carbon dioxide, lactic acid, or other cellular waste signals the need for increased blood flow to the tissues, and these signals cause capillary sphincter as well as the muscles in nearby arterioles to relax, thus increasing the blood flow to the capillaries in sort of emergency if there is increased carbon dioxide concentration, lactic acid, or cellular waste accumulated within the cells. Moreover, meta-arterioles, in case of any sort of emergency, let's suppose if there is, an inflammatory case. In case of inflammation, the blood, instead of flowing from the capillaries, will tend to flow from a meta-arteriole. So the meta-arteriole and the thoroughfare channel. From the meta-arteriole, it will tend to flow into the thoroughfare channel. Why? Because when it will flow from the meta-arteriole through the thoroughfare channel, it will cause to increase the total cross section, decrease the in such a manner that it will increase the total cross sectional area, due to which, as the total cross sectional area will be increased, the pressure will be increased, such that, let me just explain it once again here. As the total cross sectional area is decreased of meta arterioles, it will cause to decrease the pressure, due to which, you know as there is decreased pressure, it will cause to release much of the heat, because there is more of the surface area. So in this case, during inflammatory cases, there is thoroughfare channel and meta-arteriole supplying the blood. Here we have the vascular pathway. Vascular pathway basically consists of two parts: pulmonary circulation and systemic circulation. Pulmonary circulation, blood moves from the heart to the lungs, and in systemic circulation, from the heart to the body and from body to the heart. So in pulmonary circulation, it transports deoxygenated blood to the lungs to absorb oxygen and release carbon dioxide. The oxygenated blood flows back to the left atrium to the heart through the pulmonary veins, and when it comes to the blood coming from the heart to the lungs, it is done. So the transfer of blood from the right ventricle to the lungs through the pulmonary artery mechanism is previously told. And systemic circulation, the systemic circulation contains aorta and vena cava. In this type of circulation, arteries carry oxygenated blood, which is bright in red color, but veins carry deoxygenated blood and appear dull red or show blue color through skin. In such type of system, the deoxygenated blood is transferred into the right atrium, and oxygenated blood is transferred from the left ventricle into the systemic aorta. Moreover, coronary circulation, the walls of heart has its own supply of blood vessels called the coronary arteries, which supply blood to myocardium through the right and left coronary arteries. These arteries raise from the aorta just above the aortic semilunar wall, and they lie at the exterior surface of the heart, where they divide into arterioles and capillaries. The capillaries bed then enters the vein, then cardiac veins, and finally empty into the right atrium. After that, when it comes to hepatic portal vein, a portal vein system is a vascular system that begins and ends with capillary beds and has no pumping mechanism such as the heart. We have two portal systems in the body, which is the hepatic portal system. Second is the nervous portal system. It is basically not included in our book, so we are going to discuss hepatic portal system here. The hepatic portal vein, the largest vein of the system, is formed by the union of all of the veins coming from the digestive system, which includes the splenic vein, intestinal vein, uh, from the digestive system. And apart from that, within the liver, the blood flows through the series of dilated capillaries which empty into the hepatic veins. The hepatic vein leaves the liver and enters into the post caval or inferior vena cava. Enters into the post caval or inferior vena cava. Moreover, when it comes to other parts, here post caval or inferior vena cava, and that is done first transferring the blood from the hepatic portal vein to the abdominal vein, and from the abdominal vein back into the inferior vena cava. Hepatic vein leaves the liver and enters into the posterior vein or post caval or inferior vena cava. Renal circulation. The renal arteries are short and arise directly from the dorsal aorta, enter into the kidney, and gives branches which pass through medulla. Pass through the medulla in the cortex. They give rise to glomerular arterioles. From here, blood enters the peritubular capillaries and vesa recta. Uh, and you might have been known what it is all about, how the structure of nephron is formed such that the glomerulus structure is formed in such a manner that here there is a Malpighian tubules. Oops, sorry. Uh, it is basically the glomerulus, the network of thin arterioles, as well as there is a capsule which is known as Bowman's capsule. Um, both combined together will cause to give us a structure which is known as the, it will give us the complete structure of the capsule. And apart from that, the blood is drained through the veins and leaves the kidney as a single renal vein that empties into the inferior vena cava. Now, coming to other part, which is a blood pressure. Blood pressure can be defined in such a manner that the pressure which is it is a hydrostatic force which is generated by the blood against the unit area of the vessel wall, is known as blood pressure. It is measured in millimeter of mercury by a sphygmomanometer. It is generally measured on the brachial artery, but why is that so? That's because it brachial artery basically showcases uh, a brachial artery basically has the pressure of blood uh, coming into the arms, equally, as a result of which we utilize brachial artery. The reading of blood pressure consists of two numbers: 120 systolic pressure, 80 is the diastolic pressure. It is by a normal blood pressure is 120 by 80 millimeter mercury. And here, uh, when it comes to uh, you know the characteristic, it is the thing which I previously told about the our cross sectional area, such that in aorta there is less cross sectional area, due to which there is high pressure. On the other side, capillaries have high cross sectional area, high surface area, due to which they have uh, more of the due to which they have less of the blood pressure, and blood velocity is less as well. Blood pressure is high, total area is high, and blood pressure as well as the velocity of blood is less. Blood pressure is also less in the capillaries, as you can see in the graph, as well as in the aorta. The velocity of the blood is high in such a manner. It is highest in aorta, then less in arteries, and less in arteries, then least in the capillaries, and then highest in high in venules again, and then higher in veins, and then finally the same at vena cava. And same goes for blood pressure. Blood pressure will have the same sort of graph. And it comes to baroreceptors, we are going to discuss it right now. So baroreceptors are those, let us discuss that. Here it is a type of mechanoreceptors which allows the transfer of information which from the blood pressure within the autonomic nervous system. There are two types of baroreceptors: high pressure arterial baroreceptors and low pressure volume baroreceptors. High pressure arterial baroreceptors sense the blood pressure and relay the information to the brain so that the proper blood pressure can be maintained. Example of this can be found in those which are present in the carotid and aortic bodies. And the low pressure baroreceptor volume baroreceptors are found in large systemic veins, in pulmonary vessels, and in the walls of right atrium and ventricles to the heart. The low pressure baroreceptors are involved in the regulation of uh, blood volumes. Apart from that, cardiovascular diseases are those in which we are going to discuss, diseases which are relevant to heart, blood vessels, and blood circulation are known as CVS disorders or cardiovascular diseases. After that, thrombus. Thrombus is the first part. Thrombus, what is in it? An actual thrombus can be, somehow it can be considered as a symptom of disease, because there's many of the cardiovascular diseases which are relevant to thrombus, so we need to first understand what this terminology actually says. So here, when there is a clotting of blood in the blood vessels, it is known as a thrombus formation. It is known as a thrombus formation, and the main cause of this thrombosis is atherosclerotic plaque. That atherosclerotic plaque usually involves the patches of several fats in the blood vessels, and these patches damage the inner layer endothelium of the blood vessels. Then in the damaged platelets, then in the damaged region, platelets are deposited, as a result of which it leads to blood clotting, and it causes to block the passage of the blood vessels in such a manner that when the patches causes to destroy the endothelium of the blood vessels, it causes the uh, platelets to accumulate over that site of damage, as a result of which those platelets cause to form several blood clotting, as a result of which blood is clotted at that moment, and that leads to a condition in which the blood supply is blocked to that particular organ. And here this leads to a severe condition. It reduces or stops the blood supply. Food and oxygen cannot be supplied to these parts properly, and function of such affected organs is deteriorated. When thrombus occurs in coronary artery, heart attack or heart stroke occurs, and the person may die. So it is also known as coronary thrombosis, in which a heart attack occurs, heart stroke occurs, and the person can eventually die as well. Eventually not immediately die, because in coronary artery that can be a serious case. When the clot is separated and transferred in the bloodstream, it is called embolus. It is called embolus. It can damage any small artery, and such as coronary artery, it may cause angina pectoris or heart attack. Blood clot is separated and transferred in the bloodstream, it is called embolus. It can damage any small artery, and such as coronary artery, and it may cause angina pectoris or heart attack. This issue is called embolism. This can also cause to stop the blood supply to a particular organ, due to which it leads to a condition which is known as uh, hemorrhage, if I'm not wrong, hemorrhage, and stroke as well. It can also lead to stroke due to stopping blood supply to a particular organ, particularly the brain. Atherosclerosis is the disorder of the blood vessels in which arteries become hardened, such that the inner walls of the arteries become narrow, lose their elasticity due to the formation of raised patches of fats in the inner lining called atheromatous plaques. These basically cause to decrease the lumen of the arteries, and thus arteries become narrow, lose elasticity, and in such condition flow of blood is disturbed. These patches generally consist of LDL, low density lipoprotein, which consists of cholesterol and protein, 99% cholesterol and 1% protein, fibrous tissues, etc., decaying muscles, as well as some other sort of structures such as clusters of blood platelets and calcium as well. Causes such as smoking can be a cause, hypertension, and obesity, fattiness, high cholesterol level in serum, as well as uh, serum refers to the fluid which are present in the body, and severe diabetes, as well as there can be, it can some personality traits such as anxious, being anxious, or aggressive at the moment, can also lead to such condition. After that, age factor is also something which is considered. Until the severe damage to arteries and disturbance in flow of blood, there is no significant symptom of atherosclerosis. It causes pain in the chest, arms, and its jaws, called angina pectoris, right? So it can lead to a condition which is an angina pectoris, which can lead to pain in any part of the body, particularly in the heart, chest region, as well as on the back, as well as in other sort of parts of the body. Now, rest, what we are going to discuss is all about the rest. We are going to discuss about the serious cardiovascular diseases, and how we can have a treatment or surgical method in order to treat those. When it comes to heart problems, cardiovascular diseases linked to the heart are called heart problems, such as angina pectoris, heart attack, and heart failure. Angina is caused by reduced blood flow to the heart muscle. Blood carries oxygen, which the heart muscle need to survive. And when the heart muscles do not getting enough oxygen due to formation of any sort of atherosclerotic plaque, it can cause chest pain and a radiating pain in the left arm, which is called angina pectoris occurs. It is not necessary that every time it is occurring in the left arm, it can be in other arms as well. And heart attack, myocardial infarction, in which the blood vessels of heart are blocked either by thrombus or embolus, and it causes death of the part of the heart and continuous chest pain. This is known as myocardial infarction and commonly known as heart attack. If the condition persists, it can lead the person to die. And heart failure is a condition in which the heart stops working. It is characterized by lacking of the heart to pump sufficient supply of blood to the body, as without getting enough flow of blood. As a consequence of myocardial infection, there can be a heart failure, which can lead to all major body functions to disrupt, and unfortunately the person can at all can die. A successful treatment for it is heart transplant. Congenital heart problem. A congenital inherited heart problem is related heart structural problem which is presented by birth. These problems are found in the walls of heart, in the walls of heart, and in the blood vessels of heart. Example of this can be cyanosis. Cyanosis, basically it can lead to conditions such as patent foramen oval and patent ductus arteriosum. These are two conditions which can be uh, which can be the conditions in case of congenital heart problems, which lead to condition cyanosis, such that the openings, there are several openings in the embryonic, at the embryonic stage. There's form, instead of interarterial septum, there are two regions, two walls, which are known as septum primitum and septum secundum, instead of interarterial septum, which separate the right and left atrium. So uh, right and left atrium, right. After that, right and left atrium, right and left atrium. After that, these are having a gap in between them, aperture which is formed between them, which is basically known as the aperture known as foramen oval. That foramen oval, in general, it is once the fetal development is completed, that foramen oval is closed. First and foremost, why it is formed? It is formed because during the embryonic stage there is no lungs to develop, but is a non-function, due to which blood transfers directly from the right atrium to the left atrium, due to which foramen oval is formed. Now after the embryonic development, it is closed and forms two depressions that are known as fossa ovalis and fossa lunata. But in a congenital heart problem, it can cause a condition that these two the foramen oval is not closed, and this causes to directly supply the blood from the right atrium to left atrium, due to which the oxygenated and deoxygenated blood is mixed, and the newborn's blood is mixed, and thus this leads to a bluish color appearing over the skin due to the mixed blood supply, which is known as the cyanosis or blue babies. Apart from which, when it comes to uh, apart from which when it comes to another similar condition, which is patent ductus arteriosum. In embryonic stage, there is also a supply. There is also a duct which connects the pulmonary artery directly with the systemic aorta. So in this because lungs are not functional at embryonic stage, so from the systemic pulmonary artery connected to the systemic aorta through ductus arteriosum or ductus botelli, and that ductus arteriosum or ductus botelli basically being connected will it remains open in the embryonic stage, but after the embryonic stage it is closed by deposition of thin elastin fibers, which are known as the some sort of ligamentous structure, and that basically causes to form a ligamentous structure instead of ductus arteriosum, which is known as ligamentum arteriosum or ligamentum botelli. Now, in some individuals, again, there's no closure of the ductus arteriosum, due to which it remains open after the birth and again leads to the condition of cyanosis. So these are the two, congenital heart problems which are important. Apart from the principles of angiography, such that angiography is defined as the X-ray visualization of blood flow in the body, such that during an angiogram there are opaque, during an angiogram. That are areas that are opaque to X-rays are inserted into the bloodstream. The images of the path through the blood vessels can be diagnostically useful. It helps doctors to see the exact site of blocked blood vessels and also to see certain problems with organs more clearly. Treatment of cardiovascular disorders, coronary bypass, and angioplasty. When it comes to coronary bypass, it is a procedure to restore blood flow to areas of your heart. It may be required when the arteries supplying blood to heart tissue, called coronary arteries, are narrow or blocked. So in that case, we create a bypass using another artery grafted from another part of the body, usually from the chest, leg, or arm. Most of the time, we take it from the leg because there in the leg there's an additional artery. So grafting it from there and taking it from there and grafting it to the coronary artery above and below the narrowed area of blockage, as a result of which the new blood vessel is obtained, which is known as a graft, and this is known as a coronary bypass. After that, angioplasty. It is a procedure used to open blocked coronary arteries caused by pulmonary artery disease. It restores blood flow to the heart muscle without open heart surgery, in such a manner that if it involves placing a long, thin tube called the catheter into a blood vessel to place a small balloon at the site of a hindrance. When the balloon is inflated, the blood vessels should open and allow healthy flow of blood, such that here, as we are having the blood inserted into it, as the balloon will tend to expand upon the uh, you know filling of air into it, it will cause the plaque to uh, expand as well, the blood vessels to expand as well, due to the elastic nature, due to which uh, angioplasty, this leads to uh, in some sort expand the arteries, and once arteries are expanded, there's attachment of a steel, there's an attachment of uh, a ring or a grill within that region, such that that grill is what do we call it, attached, is covered with an anticoagulant substance uh, present within the blood vessels, so that it cannot lead to any sort of blood clotting in that region because of the metallic string present in it. So this is how the actual mechanism occurs. Angioplasty can be done in an emergency setting, such as heart attack. And after that, when it comes to some diagrammatic views, these are the diagrammatic views which I just explained right now. Moreover, when it comes to open heart surgery here is a type of surgery where the chest is cut down, and surgery is performed on the muscles, walls, or arteries of the heart, allowing surgeons to fix heart problems while the patient is put on a heart lung machine. It is supposed to be very dangerous operation and a very serious as well. And when it comes to hypertension, when the blood pressure is higher than the normal blood pressure, it is known as hypertension, and the person is called hypertensive. If under resting condition the mean arterial pressure is greater than 110 millimeter mercury, it is considered as high blood pressure and hypertension. In women of any age, if blood pressure is equal or greater than 160 by 95, it is called hypertensive. In man below age 45, if the blood pressure reading is above 140 by 95, it is known as a hypertensive condition. So hypertension is known as a silent killer because it affects the individual and may not show any sort of apparent symptoms until a stroke or heart attack occurs. One more thing, there are some symptoms as well, as well as some causes. When it comes to its causes are in such a manner that there is use of high intake of salt
Black
in
and white.
food, as well as uh, there can be a heredity factor, such that it can be inherited from the parents and family. Smoking can be a condition, obesity can be a condition, disorders of kidney or adrenal gland due to hormonal changes, it can lead to hypertension as well. Hypotension, on the other hand side, is the opposite, where there's lower then the low blood pressure is lower than the lower range, which is 90 by 60. If it is below 90 by 60, it is called a low blood pressure or hypertension. Symptoms includes fatigue, dizziness, nausea, and lightheadedness. uh, Moreover, when it comes to some lifestyle modification preventing cardiovascular disorder, these are to eat a healthy diet which is low in salt, saturated fats, and cholesterol, to avoid the cardiovascular disorders, as well as stay at a healthy weight, such that in order to maintain the healthy weight, the risk of cardiovascular disorder is reduced by decreasing obese condition, because obesity is one of the most important factors of hypertension, which we need to have a concern about. After which, don't smoke. We are person with cardiovascular disorder, especially, and generally a person should not have smoking, because it can not only increase our pulmonary disorders, but cardiovascular disorders as well. Increases the risk of higher blood pressure, which may cause cardiovascular disorders, as well as um, keep your cholesterol and triglyceride levels under control, such that High level of saturated fat and cholesterol can block arteries and raise the risk of cardiovascular disorder as well. So here, um, it is these are some guidelines which we need to adapt in order to resist such cardiovascular disorders. And exercise, the continuous moderate exercise keeps away from cardiovascular diseases. And these are some important factors to be considered when it comes to other part. We are now going to discuss about our final topic, which is the lymphatic system. When the tissue fluid is taken into when capillaries basically have endothelium through the endothelium, the blood, the entire blood does not passes, but the small particles such as the water and other parts of blood plasma, except the RBCs, platelets, and some WBCs, release into the interstitial cavity, due to which it forms a fluid which is known as a tissue fluid, and that tissue fluid basically has around majority of the blood amount. Now that tissue fluid being released into the blood causes to exchange some sort of gases such as carbon dioxide being released, is saturated within the tissue fluid, due to which it is released back into the uh, blood, as well as oxygen being released from the blood is then transferred towards the tissue cells. Moreover, there's transfer of nutrients as well as some metabolic waste, cellular waste, and as there's lactic acid formation. In that case, that can also be transported from one end side from the tissue cells to the blood capillaries. So in this case, as the gaseous exchange, the entire gaseous exchange as well as the material exchange takes place. Rest of the fluid which is left behind, which would be considered tissue fluid, now cannot leave back into the heart. Now, in order to drain that uh, fluid back into the heart, we need to have a particular system, a particular vessels in which we can drain the extra in this tissue fluid uh, back into the heart, because this tissue fluid is necessary in a way such that that tissue fluid, if it is not obtained, that can cause a death of a person within a few minutes. And how, let's see. So here, in order to obtain the tissue fluid back, about out of the tissue fluid around out of the tissue fluid, let me just show you over here. um yes. So here, within the tissue fluid, 85% of the fluid returns into the blood at various end of the capillaries, but the rest 15% of the tissue fluid drains into the blindly ending lymph capillaries, as lymph, along with WBCs, cell debris, and microorganisms like bacteria, are transported back to the heart through lymphatic system. Thus, lymph can be defined as a colorless body fluid that contains lymphocytes, a granular WBCs, small because they are smaller in size, uh, small proteins and fats, and lymph takes fluid substances from cells of tissues and intercellular spaces which cannot penetrate the blood capillaries. It is a medium of exchange between the blood and the body cells, which we need to body cells, and it is, in other words, can also be called as the drainage system. And apart from that, it consists of several lymphoid tissue structure known as primary lymphoid structures and secondary lymphoid structures. Primary lymphoid structures are the lymph nodes, uh, whereas secondary lymphoid structure are spleen, thymus, tonsils, as well as some patches of tissues in appendix and small intestine are also present. Uh, Lymphatic vessels for all the toxic materials in the body, which is ultimately exerted in the form of urine. And all the body tissues are bathed in a watery fluid derived from the bloodstream. We have previously studied that. And here, when it comes to the lymphatic vessels, these capillary form a complex network or microscopic narrow-walled vessels, which the substances can be moved readily. And lymph capillaries walls are components of cell walls. Of I mean components of cells. Uh, oops, components of cells with opening between them that act as one-way valves. They have one-way valves as well, which prevent the backward flow. And due to the motion, due to the valves, as well as the contraction of skeletal muscles, as well as some of the breathing movement. The flow of lymph in the lymphatic vessels is gradually able to occur. These openings allow relatively large particles along with fluid to be carried in the lymphatic capillaries. Then materials collected by the lymph capillaries flow into large lymph vessels through which lymph are forced as blood through the veins. Large vessels are somewhat, muscular walls, but most of the momentum of lymph walls comes from the contraction of nearby muscles, such as those used in breathing and walking. And as in the blood veins, direction of flow is regulated by the one-way walls, as previously mentioned, and efferent lymph vessels merge before entering one of the two ducts, the thoracic duct or the right lymphatic trunk, and the afferent one which are coming out of the thoracic duct, such that uh, you know thoracic duct or right lymphatic duct, and all of the blood now, all of the lymph from the thoracic duct, the right lymphatic trunk opens into the thoracic trunk, and from the thoracic trunk it directly opens into a structure which is known as the subclavian vein, innominate vein, uh, right subclavian vein or innominate vein. And here, when it comes to the role of lymph vessels in villi, it is previously discussed that the small intestine is richly supplied with lymph capillaries. After absorbing digested fats, intestinal cells release fat globules into the intestinal interstitial fluid in the form of chylomicrons, and these globules are too large to diffuse into the blood capillary cells. These blood globules may take up 1% of the lymphatic fluid. And apart from that, lymphoid tissue here, it is lymphoid tissue are of many types. The one which we are going to discuss here is all about the is a lymph node, which is a primary lymphoid tissue. The large lymph vessels are suspended periodically by kidney or bean-shaped structures about 1 inch long called the lymph nodes. Lymph is forced through the channels within the nodes that are lined with masses of macrophages. And as you can see, the lymph vessels which are coming inside are known as the efferent vessels, and those which are coming inside are known as afferent vessels, and those which are going outside is known as efferent vessels, such that afferent vessels are multiple, whereas efferent vessel is a single larger vessel as compared to afferent vessels. And these vessels are having several sort of capsules. These capsules are basically having masses of macrophages for the protection, and this is trabeculae, and the structure is quite similar as compared to spleen uh, in this case, as well as cortex is there, follicles are there. In such a manner, we are having the structure of kidney, we are having capsule over which there's cortex and medulla, as well as there's medullary sinus, hilus, through which the structures, efferent lymphatic vessel leave, as well as several nerves and uh, blood vessels come out and go out. So this is how the structure is formed. Lymphocytes are also produced in the nodes. Both of these white blood cells recognize and destroy foreign particles such as bacteria, virus, and are killed in this process. One more thing, that spleen is having a similar structure. It is located first and foremost, it is located on the left side of the abdominal cavity between the stomach and the diaphragm. The spleen filters the blood, and the splenic vein is having the maximum amount of hemoglobin content within it. Uh, Another MCQ's point, and spleen is also known as the uh, during the fetal development, it is responsible for development of production of RBCs within it, and spleen is responsible for filtering the blood. So these are known as the RBC, uh, blood filters. Spleen, liver are collectively known as the RBC, the blood filters of the body. So exposing it to macrophages and lymphocytes that destroy foreign particles and aged blood vessels. And here, this is an end of our topic, which we completely discussed within the past 2 hours, and hopefully we'll meet again in another video.